Is this recording?
Yes, do you mind?
No.
Okay.
Does it have a camera on it too?
It's a video recorder.
Oh, okay,
yeah.
Interesting. Alright. So basically, hopefully we'll be able to get through all of it this morning. But basically the first part has a little introduction, and then you're going to just observe the roly-polies for about 10 minutes. And so essentially you're going to separate them out of their plastic containers and then put them in little glass beakers and then watch them for about 10 minutes. And just notate what you see. Like we've already mentioned, things like, do they clump together, or do they scurry about? Some of them might be trying to climb up the side of the glass. Anything that you observe, just go ahead and notate. And y'all can write on this so that this is your copy.
Oh, okay.
Yeah. And then what you're going to do, the focus of this particular lab says it's about orientation, like do they run towards each other, away from a stimulus or whatever. And I really don't want us to get caught up in that too much. But basically what you're going to do is, you see here that we're testing where it says introduction on page S3. It says, in this activity you will observe isopods as they respond to humidity differences in their environment over time. So we won't be able to measure how much humidity, but what we typically do is we take one of these are actually called choice chambers. What a name, huh? I think it's because the roly-polies are making a choice. You're giving them a choice. Okay. So what y'all will need to do, and it's kind of tricky, I need to get a template, but I don't think I saved them from when we did this last. So what we'll do is we'll separate out the filters, and what you'll have to do is get like a pencil, and I think we've got some up there if y'all don't have one. And it's kind of tricky, like I said at first, but you want to trace and kind of make a little template with the filters. So if we get a piece of paper, we can do that, and then we'll get a more firm piece of paper once we've got a good good first filter. We can trace it on a cardboard piece or something. But anyway, so we'll try to make it the same size as the chamber, and so you'll cut out your circle, one on each side, and this also gives them a surface to walk on, so they don't just walk through the water and maybe not have good good friction between their feet and the water. But essentially what you'll do is you're going to have the filters, one on each side, and then there's these little amber water bottles that say tap water on them. So one side you're going to leave just the paper, and the other side you're going to add water to. Okay? And so you'll want to make these as flat as you can in the bottom, so you kind of want to make as best of a complete circle as you can, and then they can just freely walk back and forth between the chambers. So the way this one is set up is it has you record every 30 seconds, but honestly I think once a minute is sufficient. Okay, so for 10 minutes you're going to record the wet side versus the dry side, how many roly-polies are on each side.